Welcome back, everyone. We are, oh, gosh, do I know your information anymore? I'm all prepared here.
Everybody knows.
Everyone knows who this person is. So I'd like to, of course, introduce Dr. Awad, who's going to be speaking to us a little bit about clinical trials or the future of clinical trials, but he also wants to share just some wrap-up information from the scientific meeting that was just held, he told me. So have fun.
Hi, how are you guys still going? I know, so it's been long few days, but I know you guys look forward to it. First of all, I really have to say, just give you my deepest thanks for the very touching tribute that you guys shared with me today, and seeing the faces of the patients is what it is all about. When I went back to my room and I was speaking with my wife, I told her, you know, that about almost 40 years ago when I decided to go into medicine, the reason was to share knowledge with patients, to share my hands and skills with patients, and also my heart and caring. And you know, what I saw today is a convergence of all of that. So it makes me at ease with what I've been doing, and I thank you for really, you know, helping me stay on the road that we all want for all of us. So that's been great. What I think I do the best with you guys is try to translate some information from the science to what it means at the patient level, and maybe that's what we need to kind of spend a little bit our time. The assigned topic is the future of clinical trials, and I think that's a good topic. So I want to give you just a few thoughts about what the biology is telling us, and then what these different trial designs and trial phases and stages, because, you know, you heard about it from different people, platform and phase 1 and phase 3 and first in human and whatever. So I'll try to kind of give you a little bit of a sense, because we always think about that, and I think that's very important. And then the rest will be your questions. So, you know, you stayed here long enough to stay into the session, so you deserve to ask the questions you want, and that is good. So I think from the biology of the disease, we used to think of CCM a little bit simplistically as you inherit one copy of the gene if you have the familial disease, and then the second copy is gone and you form a lesion. So the lesion has both copies screwed up. And that's why you're not all a lesion, because, you know, the rest of your cells have one good copy of the gene. So that model is very good, but it fails at some points, because even the humans who get many lesions, some lesions are big, some lesions are small. Some bleed, some don't bleed. So it's not like all of them form because of the same thing that happens and they behave the same way in a predestined. So there is enough variability, and there is enough variability among families, and that shouldn't really happen. If everything just happens because of a gene, second copy goes bad, then it should all be statistically similar. So there are disease modifiers we learned about, and some of them are genetic, and the best way to look at those genetic ones was to really look at families that all have the same mutation. So that's why the New Mexico group was studied very extensively, to try to see within that group who all have the same mutation. So they all should have the same disease. Why do some people have different ones? And we did learn that we're not born with just the CCM mutation and get a second hit, but there are other genes we have, or gene variants we have, that make us more predisposed to getting more or less lesions. And the general theme of those genes are the so-called pro-inflammatory genes. So if some of us inherently are more driven toward having pro-inflammatory activity, then the lesions form more and they're more aggressive. And this is how it fits in with the vitamin D story, where if you have vitamin D deficiency, the disease is more aggressive, and so on and so forth. Then we also learned that there are other potential modifiers. The microbiome story has an implication on diet and on whether our gut is healthy or not. And a small fraction of us families who have the disease have maybe inflammatory bowel disease or gluten allergies or other things that make the gut a little bit more leaky than the average gut. And that ends up being one of the factors that helps raise the inflammation level and drive the disease in a very, very unique way. And there are other things. You heard about the estrogen hormone supplements. We've suspected it. We've been kind of saying it during clinic visit, but telling you we don't really have a lot of evidence. Now we're starting to find evidence in the population about that, and those things will happen. So the guys say, well, what about us? We don't do estrogen, you know, but I tell the guys you also have prostatitis and other things that can cause also fluctuations in the microbiome and things like that. So all of us live in a milieu that has triggers. And some of those triggers help worsen the disease, and we're learning a little bit about that. And then the question came up as to, what about me who has only one lesion? I don't have the family problem. I've been reassured I don't have the familial disease. Then that lesion carries the exact same molecular signature as the lesion that happens in the family form. With a few exceptions, it is much more likely to happen near a developmental venous anomaly. You've all heard that. So that if you have a DVA, even a subtle one, that's where the lesion will grow. It doesn't grow elsewhere in the brain, it grows there. And that becomes the genetic primer. So you don't have the other genetic primer where every cell in your body has one copy gone, but you happen to have one vein in one area that's now the primer. So if a second copy happens to go bad, or a second gene happens to go abnormal anywhere in the brain, nothing happens. But if it happens near that vein, the two combinations, the pre-existing mutation in the vein plus the lesion, will now grow. So this information really just came up the last year. We didn't really know that. So that has been very, very important. The other thing we discovered a lot, and it came from this meeting, a lot of it, is these other somatic mutations that happen only in the lesion. So they don't happen elsewhere in your body. They just happen in the lesion. And when they do happen, they drive the lesion to become more aggressive. So one of them is this PIK3CA mutation that we think if it happens on top of a familial lesion, it could transform the lesion from a tiny little dot on SWI to being the real big cavernoma. But we don't have enough proof to know that that's the only thing that drives that change. We suspect it is, but we have a lot of work to do. We were just talking right before I came, what kept me late is, you know, if we want to look for that, how is the best way to kind of test that hypothesis and try to understand how many PIK3CA mutations do you need to have an aggressive lesion? Can you have an aggressive lesion without them? And can you have lesion with PIK3CA that's not aggressive? I think that's an important question, because if we want to select patients for certain treatments, that could affect how we select people. Or alternatively, everybody has some degree of it and therefore will benefit from the same drug. So the idea here is trying to separate us a little bit, not just based on familial versus sporadic, but to go to another level of precision medicine where you understand which lesion is now becoming what and what's driving it, you know. Some of you that have participated in the research with my team know that for a few years I've been interested in the switch that goes from cash to non-cash. So many of you tell me if I have 20 lesions in my brain and they never grow or bleed, you know, I am what I am and I'll move on. But what I dread is what's going to happen to change one lesion, is particularly one in the brainstem or whatever, into something bad. And so far, you know, we look at you blank stare. I don't know which of your lesions will misbehave. I don't even know if any will misbehave. But that's a big life-changing difference, right? And so if we understood that switch, that may not be the only switch, but if we understood that switch, we'll be able to do a lot to kind of target that switch, select the people for treatment who are destined or more likely to bleed and so forth. So there is a lot of movement on that level. And the final point I want to tell you from the biology that has come up, then I'll switch a little bit to trials, is this issue of within the lesion, there is bleeding, there is clotting, and there is inflammation. And all of them are kind of a mix of badness, you know? I mean, if you have some new blood in the lesion, I don't think you really care that much whether it's a thrombus or a blood that became a thrombus or did it leak and became a thrombus. But it could make a difference in view of how we alter the disease and how we target it. And whenever you have bleeding in the lesion, it also triggers inflammation. So inflammation becomes one that makes the lesion more aggressive, but also when you have bleeding, there is inflammation in it. So that may be one of the reasons that once a lesion bleeds, it becomes more likely to bleed until it's chilled back down. Understanding those is what a lot of smart minds in the other room are trying to figure out, and what we're trying through our biomarker discovery, try to kind of find out if we can see in the blood the difference in signatures between lesions that misbehave, lesions that have PIK3CA or not, and so forth. So do you want to take a few minutes to ask me about those biologic principles, and then I'll go to the trials part of the discussion? Yeah.
Thank you. So is there any more known about what can be done to be anti-inflammatory, or that we can do regarding that?
So, you know, those of you who have sat with me during a clinic consultation, we go over what we call the triggers. What are things that you could do that can be bad, or we think can be bad, and we go over those lists. One of them is vitamin D deficiency, because it is a pro-inflammatory condition. And there are many signals, both looking back at the disease and also in people going forward, that if you have vitamin D deficiency, you're more likely to bleed. So that's one factor that is easy to address, right? It's not that hard. You go to CVS, get the biggest jar on sale, and you take care of that problem, right? And don't think the sunshine alone will do it, because there are as many vitamin D deficient people in Florida as there are in Cleveland, Ohio. So I think it's just how our body metabolizes it. So that's one factor. But there are a lot of others. You know, we meet people that happen to be on Adderall or Ritalin for their attention deficit disorder, and they didn't realize that when they started to take it, that's when they started to bleed, until somebody put two and two together. Or birth control pill, or in vitro fertilization, which kind of is like a birth control pill big time, and so forth. So all of those are things we talk about, and they're difficult conversations, right? Because the vitamin D one is easy, but others, well, I really want Botox, Would one Botox be okay? Well, I don't know, maybe it'll be okay, but we have had patients whose only trigger was Botox, and when they had it again, they had another bleed. So I had to report it, and I have to tell you until we know better. So those are how we approach it. There is some things that are becoming more proven. Some things are a hunch, and you pay me to share my hunches with you. So that's kind of where we are, That answer your question?
Yes, and I think with also thinking about the gut drain because I think before it was like it's not known what you can do for a leaky gut. And I don't know if that's evolved.
So we have a very exciting project that's now about halfway through. So, it's really going very well, where we're collecting literally hundreds of blood samples from people who either bled in the past year or who didn't. And then when I follow you over a year, whether you return or whether we call you to ask you, we can also separate whether you bleed in the next year or not, and to try to see what signals in the blood separate those two groups. And it's very interesting. We start with maybe about 30 or 40 reliable signals that every time we take a group, it could be a different group, or they could have been collected at Mayo Clinic or at whatever. The same thing comes up in Brazil. The same thing comes up. So there are some signature molecules that separate the people who bleed from don't bleed. And we're trying to kind of minimize them to the most reproducible test. So if there are like 3 molecules or 4 molecules that we could look at that reflect that state, that would be the blood test for angry cavernoma or cash versus a non-cash cavernoma. I think that would be transformative in the field, and as other things also happen, because it will help you with the decision, because there are certain things you can do and certain things you don't know.
This is more of a genetics type question. Hi, Dr. Awad.
Sure.
Hi. I am curious if there's any connection with either bleeding in the cavernomas or the deficiency of vitamin D with the MTHFR gene, or if there's been any association with people who have that gene and a CCM.
It's a hypothesis right now. It's every day there is a new molecule that we discover, we think about, or a new gene, and we have to be very careful before we do the association. And also it has to be an actionable association. It's not like you can change your genes like you change your genes, So we have to know, what the heck you can do about it, And I think we will know more in the next couple of years. I really don't have anything very firm to tell you today.
For someone with a well-behaved sporadic lesion.
Touch wood.
Yes, thank you. Is there any evidence of perhaps danger of maybe passing that along to children someday? Anything like that?
No. So that has been looked at extensively, and except for the theory that anybody can be struck by lightning twice, okay? Except for that, you're not more likely to pass it to children than anyone else is likely to have a child that has it. And I think this probably is one of the biggest things in the guidelines that really helped us, is that we can tell you that if you have an SWI MRI at 3 Tesla and you have only one lesion, there is a vein next to that lesion, and you have no family history, although 3, I have yet to see an exception to that being ever. The problem is that sometimes you have 2 out of the 3, and maybe grandma had something and whatever. Then we have to sort it out a little bit. Or I may have 2 lesions next to a vein. Are they both the same lesion, or am I familial? I have 2 lesions. But if you have only one and no family history and the vein next to it, no worries about the kids. Worry about something else.
I know it got touched on in some of the discussion here, but the whole gut health and the microbiome, and wondered where the research is going on that. And it seems like what you're learning from that affects everybody, not just the CCM world.
You're absolutely right. So first of all, gut health and pro-inflammatory conditions apply to people with arthritis, apply to people who worry about Alzheimer's, apply to a lot of factors. And what's in common is that pro-inflammatory signal. It can act differently in different diseases. In CCM, it's so real. I'm going to tell you just a couple of things you may or may not have known about. The mutant endothelium on the cavernoma is so particularly vulnerable to this lipopolysaccharide molecule from the gram-negative bugs that if you don't have the mutation in the endothelium, it doesn't matter. You can have a prostatitis and UTI, nothing will happen. But if you happen to have that bad activity in the endothelium, which we happen to have it in the CCM, that CCM becomes a vulnerable target for that particular bug juice, if you want, or bug molecule. And the reason some people are more affected than others has to do with the ecology of the gut. It's not just, do I have one bug? All of us have gram-negative bacteria. So the idea is how the ecology controlled, and how do the bugs control each other so you don't have more circulating lipopolysaccharide. And that is what we know, okay? And it is so powerful that we can have microbiota or fecal samples of people who will never get a CCM. Okay, the problem is I can't make yours like that because we don't know how to change. We can only know it as an explanation, but we cannot really create the ideal gut environment that protects from CCM. If we do that, I think, that would be awesome. But what we do know is that certain things make the gut more likely to drive CCM. We talked about those, some of the food preservatives, and we tested that in the mice, and it works. That doesn't mean that every time you have a yogurt with a preservative, it's going to do it. But on the average, it's a driver. So if you can avoid the driver, it's better. That's kind of how we approach this. And it's not like you take one-time kombucha and it's going to change your gut into a raging gut. But we don't know how you can make the gut better. And I've heard so many dietitians tell you that they know, but they don't know about the CCM microbiome. They go back to our same paper to look it up, to see how their diet, that's, I can't take that to the bank, nor can you. Now, what may be a bright signal is, of course, none of us wants a test where you give a fecal sample to know how is my feces doing now, or they have enough good bacteria. Although you do it, but it's hard. In fact, the way we find those differences is something you may not have thought about, but the way we find them is by taking a group that has something and a group that doesn't, and looking at very advanced computer techniques that look at the genes of the bugs, of the group that has it or the group that doesn't have it. So if I have yours, I don't know where it's going to fit unless I look at the whole group. And that's not good enough for a biomarker. For a biomarker, you need to have something in the stool signature that says, my stools are pro-cavernoma or anti-cavernoma. So what the specialists in the gut are telling us is that you're not going to get it from the feces smear because that's so variable and so forth. Where you're going to get it is potentially from molecules in the blood caused by the bad microbiome. And we've been, very aggressively looking for that. These are metabolites. They are not the inflammatory proteins. They're little metabolites that have to do with the liver and with bile and so forth. But they reflect different status of the microbiome in the gut. And if we throw in one or two of those molecules in the blood test of cash versus non-cash, that may capture the microbiome contribution. And that's kind of where the thinking is. And if it sounds hard, it is hard. It's not an easy, or we would have already solved it for you. Any other questions before I switch to trials?
Hi, Dr. Awad. I'm so happy to be here. I think you're wonderful. And I'm not trying to pump my question, but I just want to say that I wasn't on the video, but I think you're amazing. So thank you so much for the work that you do. I wanted to hit a little bit more on the leaky gut issue. I know we've talked so much about emulsifiers as contributing and making that, the endothelium more vulnerable. But people who regularly have a leaky gut, there's certain diets that they're recommending people to have, and taking different supplements or just the diet you have.
Yes.
Is there any connection of us who have CCMs to follow a leaky gut diet in addition to having emulsifiers?
Yeah, so the problem is we haven't had studies that looked at whether leaky gut diet makes up for emulsifiers or doesn't. And the only reason we mention emulsifiers is that's the only factor that we did test. There may be others. It is more of an unknown problem. So I turn around the question and I say, can you be sure that what you're doing to your gut is making it better? You see, so that's where I come from, is the idea. I mean, everybody wants to throw something at the gut to make it better, but do they know? And not just better gut for general gut health, but better for CCM gut, which may not always be the same. Now, I think if somebody has inflammatory bowel disease, they should get on biologic. And we have evidence that B-cell depletion, for example, helps CCMs. So I tell the people who have rheumatoid arthritis, gut leaking, ulcerative colitis, etc., don't mess with therapies that you don't know what they're gonna do. Go treat your disease, and it'll probably help your CCM, and that's kind of one factor. But when it comes to what do I eat to make it better, I don't know if feeling better from your gut means it's gonna be better for your CCM.
Thank you for that. And I was wondering, I've seen a few things pop up in my social media feeds lately about gut microbiome testing, and that you can send a sample of whatever, and they're going to tell you about your gut microbiome. And I've seen also like gastroenterologists offer that type of testing. Is that anything
it doesn't help. Okay. So I wish it would have helped. I would have loved to go to the FDA directly with the gut microbiome that's bad for CCM and make that the test for all of you. That would have been the easiest route. But what we've learned from studying this is the difference that drives CCM can only be detected in groups of patients. Therefore, the test of one smear of feces ain't gonna do it. No problem. All right, let me switch a little bit to the trials world, because that's just as confusing, right? I mean, phase 1, phase 2, what do I do with? If you think it's good, why don't you give me the medication, etc.? And what medication is good enough to test, and in whom, and so forth. And there are some general principles. The very, very simple, safe, low-risk medications are probably going to have a very small effect. Like vitamin D, if everybody does them, if we have $10 million in the US to do trials for cavernoma, I wouldn't spend it on a vitamin D trial. I just tell you, take the vitamin D, get that out of the way, let's do better with other things. Aspirin is another one of those, and the association with aspirin is very controversial, simply because I tell people, if you need aspirin for your heart, take it. If you don't need aspirin for your heart, is it really better for your CCM? We don't know. And after you bleed from your CCM, you probably should hold the aspirin for a little while. So everybody agrees on that. But what that means is that when you take a patient who bled or didn't bleed, and you find that those that didn't bleed were more likely to be on aspirin, is that the chicken or the egg? since I already tell you to stop the aspirin because you bled. So, or you told yourself to stop the aspirin because you bled. So you're more likely to have had bleeding despite that. And so we haven't had controlled trials. So the only way to do this is to actually have a group of CCM patients take aspirin all the time versus not. But then look what happens. I'm not advocating that trial. In fact, Connie knows that's not where I want to push the money to go. Because, you know, what if you bleed in your lesion? You're going to stay in the trial, or you're going you know, you're gonna probably stop the aspirin right there and then. And therefore, it probably wouldn't change what we now do, you know? I mean, if you need it for your heart, take it. You know, if you want to take every other day aspirin because you think it's healthy, take it. But I don't think we can tell you that's going to change black and white your CCM behavior. And if you have had a recent hemorrhage, probably hold it until the lesion stabilizes. So I think, you know, there are other questions where we could make a bigger difference, and some drugs that can make a bigger difference. So, you know, the Rho kinase inhibition is a whole class of drugs. We're starting with testing the statin for its Rho kinase inhibition, but there may be drugs coming out from Pharma that you heard about that are designed to target Rho kinase without any of the side effects of statin. And they may even be more of a brain-specific Rho kinase inhibitor. So those could carry a lot of promise. But what we do not know, those drugs have not been given yet to humans. So are they going to grow horns on us? I don't know. We have to go through the steps. We have to go through the steps. And there are other drugs where we know, like the temporal drug that's being tested by Recursion, that it seems to be safe. People who take it are not having side effects, but none of the trials currently are designed to look for a benefit. So you're in the trial, you know, fine, get in the trial. It's not a problem, but there is zero chance of getting a signal benefit at this phase. So the trials go in phases. There is a phase 1, which is safety. Phase 2A still targets safety as long as the drug is now past phase 1, meaning statin doesn't need phase 1 because it's already been given to gazillion patients. So we're able to combine phase 1, 2A together. So give it to CCM and see if it's safe in CCM. But a drug that's new, like Tempol or the drug by Norelis, is going to go through a phase 1 in CCM to make sure we're not doing any harm before we move it. So those are, when you hear those terms, that's what they are. And you can speed those phases up, and believe me, the pharma want to speed it up. That has to do with the value of their drug. So you can't rush them more than what they rush themselves. You know, they're in there to make the money and to get this drug to win, which is good. And this is why we need to have them bring the money to our disease, because the NIH operates separately. The NIH will work on previously approved drugs like rapamycin, statin, that are no longer owned by a pharma. There is no patent on them anymore. And therefore, propranolol is one of them. So those are the type that the NIH will do trials for. But of course, if a big drug comes with 10 times the money being thrown at it by pharma, then the NIH trials will lose in the process. So it's a healthy competition, but we have to kind of be careful, and so that we give all the possible beneficial drugs a chance. We don't put all our eggs in one basket. And the leadership of this group is very knowledgeable about this. I don't have to tell you this, because you're actually smarter than any of us on that. And you know that what we're going to do here is not we're putting all our eggs in one basket. We're going to try to see what is promising, that has some money and somebody after it. And as long as it's safe, we're going to try to see if it works or not. And we're in a better place than we were 5 years ago, right? We were dreaming about trials. Now we're talking about which trials we're going to do. And you as individuals have to first ask yourself a question: why should I participate in a trial? And we face this all the time. And The alternative is to start picking at using drugs like snake oil. So somebody tells you, oh, this one may work, why don't you try it? And N of 1 never proves anything, you know? You may take it, then if you don't bleed, you feel it works. If you bleed, you say, oh, I made a mistake. But that's not how it works. The way it works is we have to do a lot of groups, and you have not to know whether you're taking the drug or not. Otherwise, you figure out by bias what's going on. This was our biggest critique of the Italian study. They told the people, you're taking placebo or propranolol. So the one taking placebo never worried about dizziness. The one taking propranolol, every day they felt dizzy. You know, because that's what, you know, so when you're blinded, I think it helps us be a little bit more objective. And it's the fastest way to get to an answer, so that after that, we can tell you honestly, forget it, this is BS, doesn't work. Don't waste your time on it. And if you say, what's the harm of trying, that may be okay for a vitamin D where there is no consequence. But it's not okay when you start taking drugs that may have side effects or may be harmful to CCM. And they haven't been properly tested. And therefore, you know, you think you're helping yourself, but instead of actually enrolling in a trial where you might get a good drug, you chose a drug that's hogwash. Okay, and this is where I think the issue. Now, you will have choices of drugs with varying known or unknown side effects, and that will be appropriate for choosing what trial you wanna go into. We have patients who have had huge repeated bleeds in a child, and they've been put on drugs you haven't even heard about, things like angiogenesis inhibitors or high-dose rapamycin, but for one case, and then if it stopped bleeding, we don't really know that that's what did it, and most of you don't have that problem. You know, so you can't take what we did for that kid and apply it for your lesion. So the benign disease versus aggressive disease is going to be a little bit different for the trials. And this is why kind of take everybody and give them the same drug is probably not going to work. So we're going to have to, at some point, start to say, for this trial, we're going to take the patients that have bled, therefore they have more likelihood of bleeding, and maybe you know it's justified. But for this other drug, everybody should take it, you know, like vitamin D and so forth. Does that help a little bit understand where the Connie, is that how you see it? Because you know you've been sitting through these same discussions. So.
Yeah. So yeah, I think one of the things that you need to talk about is platform trial, because I think that becomes important as we try to think about the timing of trials, because at this time we've been, for the most part, doing one long trial, and then we may start another long trial, but there's an option.
So let me tell you the two trials that have already played out in this field, with 3 trials played out in this field, and the lessons learned from them. The first trial, everybody jumped on it, which was to give low-dose simvastatin to people with familial disease in New Mexico. You remember that? And you know, anybody who objected about that trial, it's like you're against apple pie and Chevrolet. You know, yeah, better sign up for it. So what happened there? So they from the beginning said we're only going to test like 20 patients. And then when they did that, and the dose that was chosen had no biologic premise of doing anything. And therefore, they did the 20 patients, and no difference was shown, except that the simvastatin was reasonably tolerated. Nobody quit because of it. Does that help you? Now, nothing was bad there, because, you know, it didn't like catch the whole population of CCM and froze them into that trial. It was only a small trial. But then there was the Italian trial and our trial, and both of them have issues. So I'm gonna first address ours, because it's easier to self-critique. So ours, we chose atorvastatin, at a dose that is high enough to cause the Rho kinase inhibition. And we chose sufficient number of patients where we have a two-tailed effect. This is a very, very difficult high bar to show a benefit on even microbleed in the lesions. So we're not waiting to see whether you're going to have a big bleed. We hope not many people, there are only 5 in the trial, have had it, and you're not going to have that big a difference there. But if we saw that the lesions are becoming more leaky or less leaky with the drug, then it gives us a greater firm foundation to take that drug and tell a bigger CCM community, let's do a real big trial, even if it takes all of us getting into it, because there is enough signal of benefit. So what's wrong with this approach? Not much, or I wouldn't have done it, but the main approach is speed. It's agonizingly slow. It's agonizingly slow. So the measuring of leak, when we started the trial, could only be done at our institution. Now we have taught a number of high volume centers, UCSF, Mayo Clinic, Barrow, they all know how to do it. So the next trial doesn't have to be only at the University of Chicago. But when we started, those were the tools. So it was agonizingly slow. Now we say, okay, why don't we put rapamycin and do the same type of thing, but doing twice as fast and get that answer and see if rapamycin is the answer. The only problem that I have with that, and I would love to do it, and we almost are ready, we have everything together to do it, is that even at the fastest speed, it's like 3 years probably at multiple centers to complete it, as opposed to 5.5 years, which we had with the other trial. 3 years is still slow, and the other problem with this is that I don't know how you as a community are gonna embrace a drug that 20% of you will have diabetes as a result of it. 15% will have some issues of mucosal ulcers as a result of it. Now, it may be justified to prevent a brainstem bleed, but most of you are not bleeding every day, and you're because I've talked to a lot of you, and you even have concerns about taking the damn statin. So the issue there becomes, so this is where a potential solution is this platform trial, where nobody has to carry the guilt alone, the responsibility alone. We have it across a big population swath, so there'll be CCM patients from as far away as Italy and Finland and England and the US and Canada and Australia. So strength in numbers. And then we can try 3 or 4 potential drugs that have benefit with sharing one placebo. So in the US, you may even choose which one you want to be into. And that's a nice thing. You say, I want to get in, but I only want to be in this limb. And that's enough as long as we balance it across the platform. So not everybody has to be randomized to propranolol or atorvastatin or whatever. So that has an advantage. But like with everything, it's, it looks better until you start looking at the details. So we're struggling with the details, So how do you let the drug into this trial? What are the criteria? Just some benefit in a mouse? Is that enough? Or, how safe does it have to be shown? Therefore, does it need a phase 1-2A before it gets into that? Or does it have to have been approved for another indication? Therefore, we know it doesn't make you grow horns. And so it may be appropriate for those repurposed drugs where no pharma is behind, right? And, is propranolol better than statin? Is rapamycin even better? Or should it be taken or not? We could answer some of those questions with strength and numbers and do them all within a 5 to 8 year span instead of 3 years, and then I start again. And oh, I didn't give you the example of the Italian trial, which was just published. And 3 years, it was fast. It was not 3.5, but it was designed from the very beginning with such low event rate that there was really no chance to show a difference. So the trial is done, and they had 4 bleeds in one group and 2 bleeds in the other. Are you willing to believe that that's real? So in fact, I was asked to write a commentary, and the title of my commentary was Credulity. It's like strange credulity. You can't, now you're going to ask everybody in Europe to go into a propranolol trial based on that data. So that's not enough. So that won't happen in a platform trial. The platform trial will have rules whereby, you don't, throw away a drug or adapt it unless you have some very strong signal. So our thinking in general, you've heard from a lot of people who are advocating different trial designs, is that maybe these drugs that are not owned by a particular pharma can be put together in a platform trial, and then let pharma bring the other drugs to the table, one at a time, the way they have to get them approved by the FDA. And you'll have choices. Do I want to? And maybe one will only enroll cash cases, while the other will enroll any CCM and see what happens. And that will give some choices for different groups of you to be involved in different trials. It was very disappointing to many of you that the Atorva was only open for cash cases. Why? That's because I wanted to have enough bleeds to tell the difference. If I gave it to everybody, I'll be where the Italians are today. Can't tell nothing,
And one other thing I wanted to add to that is just because it's a platform trial and it sounds like it's just for drugs, that's not necessarily the case. It can be for other types of interventions too. You guys heard today about focused ultrasound. I don't know that that's going to lead, that research will lead anywhere, but if it does, there's no reason that a platform trial couldn't include that type of an arm as well.
Yeah, now the methodology has become very complicated because the candidate for surgery versus no surgery is different from the candidate to take propranolol, yes or not. Because you may be willing to take propranolol no matter what and hope that it works, versus you're not going to let somebody crack your head unless you're at a different level of risk. So they're not all the same, and the platform trials will start running into that. There are other problems with platform trials that probably will be the issue, which have to do with who funds them. So when they are across international platforms, the US government will do some international trials that are for the benefit of US patients, and we may push our senators to help us with that. But if the majority of enrollees are going to be outside the US and the data center and the big scientists analyzing it are in Scotland, I don't think the NIH is going to put US federal money into that. So we have to find creative ways, and we've been sitting at the table thinking of how do you create a data center for American patients funded by the US government, a data center in Europe, and maybe the rest of the world funded by another conglomerate, but with upfront synergy of sharing data. So we can sell that easy to the NIH. I mean, I sat on the NIH Council for 4 years, and they are very receptive to telling them our patients will benefit by sharing data. And that will be fine, but they don't want to send all $40 million of US money to be done, in China or in Australia. They're not going to fund that. It's not real. So at the end of the day, and conversely, the same. The UK government is not going to fund patients being enrolled in trials in the US. So we have to find a way of how the sum is bigger than the part. And the good news is that everybody who really is involved in this are sitting at the same table. And this is not the case with many diseases. And if there is something that the Alliance to Cure has done, and its models around the world, is this idea of converting stakeholder competitors, whether they like it or not, into collaborating. I think it's a great thing. And we really sat this morning and started to sketch how do we really push this so we create a model that works to be able to drive that. We'll see. We'll see if that's going to move forward. We have a year to plan it, and if at the end of the year it looks like a plane and it looks like it's going to fly, it may fly. If it doesn't, then we'll tell you that didn't fly, but we have other alternatives.
Okay. I want to see before we end if there are any questions. Kevin.
And thanks for everything you're doing, Dr. Awad. It's amazing, all the research. And I treat kids with infantile hemangiomas, and within a couple of weeks, you usually see an effect with the propranolol. And so my question to you is, for CCM, what would be an effective time frame that you would expect to see some kind of change on your drugs? And how would you, if you could do an MRI every week, I mean, what do you think?
But the problem is that, unlike the infantile hemangiomas, the rate of regression of CCMs on any of our best drug is nearly zero. We basically are aiming for them not to grow. So then it's different already from infantile hemangioma. And then, I recently reviewed a paper on infantile hemangioma, and they have a baseline growth rate in the control group of 47%. Okay, now you can target that, right? But if your baseline growth rate is a 3% event rate, like in the Italian trial, good luck with that, man. So it's statistics. So then how do we select the group that has a 15-20% chance of bleeding again, unless you go into their brain? I think we have this group within our community, and maybe they have to be the first that we step forward and do some trials in them. Now, once they work there, then generalizing is very easy. Then every patient will say, I'm willing to take this drug. But you have to first know whether it works. Otherwise, it's going to lock you into something useless and prevent you from doing the next best thing, which is a drug that may work. So that's the other problem. Thank you so much. And really this type of discussion, and thank you, Connie, for allowing me to do this once in a while. And I will continue to do it because, just stepping off from the leadership of the Scientific Advisory Board doesn't mean I'm not taking care of CCM patients every day. It doesn't mean I don't love you just the same, and that my life's work remains this, and I have a lot more to do. So we're going to keep talking, we're going to keep doing, we're going to keep working hard together. Thank you.
Thank you.